yesterday I was actually talking during the stream, but nobody could hear me because when I added the video, uh, it for some reason changed my microphone settings. So I kept talking yesterday. If you're from yesterday, sorry about that. Um, so tomorrow is my final exam for biology 2. Um, I had never actually intention to do like videos of myself with my streaming, but um, I'm having a really hard time focusing on getting things done. I had been behind because I had COVID. I'm almost entirely caught up now. The day before finals, great. So the goal today is to finish chapter 29 and chapter 30, which might take a little over an hour, and then I have to transcribe. So there's probably going to be, when I say transcribe, I mean make flashcards. So tomorrow I'm planning to just spend the day up until the last second cramming with my flashcards. Most of my exam is going to be on Unit 1, so there's part of me that like doesn't even want to finish this part because I could spend a lot more time reviewing part 1. I did fail the third exam though because I wasn't done. So kind of want to finish, but yeah. So that's the general plan. I'm using video this time though, and kind of being live because it's holding me accountable, I guess, like, because I know people could be watching, so I have to stay at my desk. So that's the logic behind that. Anyway, let's get started.
Receptors membrane, and as we'll see, it's ions flowing across the membrane via ion channels. So now that we kind of have these two steps down and some of the in perception, and those steps are reception, transduction, transmission, and processing, which we see on this first bullet point. So we're going to start off by looking at reception. Stimuli are received or detected by sensory receptors, which are special types of neurons that respond to or receive sensory stimuli like pressure, like chemicals. So like our eyes, for example, contain sensory receptors that detect light. And we saw in the cow eye dissection that the retina contains photoreceptors, which are the specific type of receptors that detect light. So these sensory receptors react to a stimulus in the environment and send information to the brain for processing. And in a few slides, we'll kind of learn about the different types of receptors and the different ways that these 4 steps occur. So in all animals, sensory receptors cause ion channels to open or close. This results in a flow of ions across the membrane, which we know alters the membrane potential. Remember that membrane potential is the electrical charge difference across the plasma membrane, and the membrane potential changes in response to sodium ion influx or potassium ion efflux or things like that. So the reception of a stimulus, of a ligand, whatever it might be, leads to the next step, which is transduction. So with signal transduction, we see the conversion of the stimulus to a receptor potential. What I mean by this is that the external signal is transduced into an internal signal. So the binding of, let's say, a ligand to its receptor results in a change in the membrane potential inside the neuron, inside the sensory receptor. That's the particular neuron that we're looking at, a sensory receptor. So if we look at this picture, we see that when a stimulus is received by a sensory receptor, it results in a signal transduction pathway being activated such that the external signal, which in this case is a sugar molecule, is converted into an internal signal, which is changes inside the sensory receptor cell. So we have our ligand, sugar molecules binding to the receptor, converting it to an internal signal, and we can see here that transduction results in ions flowing into the cell. So we see ion channels opening, things moving into the cell, a change in the receptor potential. So in the second step, in transduction, there's a change in membrane potential due to ions flowing in. This change in membrane potential, in this particular case, in this particular type of neuron, in a sensory receptor neuron, is called a receptor potential. So it's the same thing as a membrane potential, where we're looking at a difference in charge across the membrane. We're just specifying the type of neuron we're looking at, the sensory receptor. So the receptor potential refers to the difference in charge across a sensory receptor's membrane. And as we'll see, it's ions flowing across the membrane via ion channels. So now that we kind of have these 2 steps down and some of the terminology down, we're going to look at the 4 steps of perception by looking at what happens in taste. So as we know, taste is one of our senses, and what we're going to look at now is how do we eat something and determine it is sweet. In taste, the stimulus is a chemical. We're going to specifically look at a chemical like sugar, like glucose, as our specific chemical. So the receptor for taste stimuli is the taste bud, which we see pictured here. And the taste bud is really made up of several types of receptor cells, several types of specialized neurons. And that's what we can see here, sensory receptor cells, different ones that make up the taste bud. So the external ligand, the external signal, the sugar molecule, this chemical, binds to the taste receptor cell. The signal is received, that is the ligand, the external signal, the sugar molecule, this chemical, binds to the taste receptor cell. The signal is received. That is the first step. That is reception. This, in turn, causes changes inside the cell that result in transduction. So the signal transduction pathway results in sodium ions flowing from high to low into the cell, sodium ion influx, because sodium ion channels are opened. As we know, this influx of sodium ions causes a change in membrane potential, a change in the receptor potential. This is a depolarizing signal that moves the neuron from its resting potential of negative 70 millivolts to a more positive voltage. That should all be familiar from the previous chapter. So this change in voltage with the inside becoming more positive can lead to the third step, which is transmission of signals from the receptor cell to the central nervous system. So in the second step, transduction, we see a series of internal changes. We see a change in the receptor potential due to ion flow. Then we see a change, that change in the receptor potential results in neurotransmitters being released from the receptor cell into the synapse. Neurotransmitters diffuse across the synapse to the postsynaptic or receiving neuron, which is called a sensory neuron. This sensory neuron responds to the neurotransmitters by sending action potentials along its axon and all the way to the brain. So the sensory neuron transmits action potentials to the brain. You can see here that the sensory receptor cell synapses with or releases neurotransmitters to a second neuron, the sensory neuron. And that sensory neuron is the one that extends into the central nervous system. It's a neuron that carries a signal to the brain, and it's when the second neuron fires action potentials that we're in that third step of transmission. Remember that the frequency of action potentials is correlated with the intensity of the stimulus. And so a lot of receptor cells are constantly secreting a baseline amount of neurotransmitter into the synapse. And to understand what I mean by that, we need to look at the figure at the bottom of this slide. So in this figure, we can see that when there's no sugar present, the sensory neuron is sending action potentials every so often, pretty infrequently. That's the baseline condition. However, when there is sugar present, the sensory neuron sends action potentials frequently, and we see way more action potentials. Notice there's that peak and then the drop. You see depolarization, repolarization, hyperpolarization, resting potential, right? That's representing the action potential. So with no sugar, very infrequently. When sugar is present, much more frequently. So the stronger the stimulus, i.e. the more sugar present, the greater the receptor potential, the more neurotransmitters released, and the more frequently the sensory neuron transmits action potentials to the brain. So the intensity of the stimulus is coded for by the frequency of action potentials. It's coded for during the transmission step. The fourth and final step is processing, which occurs when these action potentials reach the brain. So the brain interprets the intensity of the stimulus. So it's like, this is not very sweet, this is very sweet, etc. The brain interprets the intensity of the stimulus from the rate at which it receives action potentials, like I said before. There are a lot of neurons in the brain that process these signals to generate our perception of the stimulus. And one particular area is called the gustatory cortex. So let's kind of look at the processing step a little bit more here, since we are familiar with some parts of the brain and their respective functions. So this is a picture of our head showing how information from our tongue travels to our brain for processing of taste. So taste receptors on our tongue deal with perception. Voltage changes in the receptor cell and the taste bud result in neurotransmitter release. Into the synapse, that's transduction. Sensory nerves carry that signal to the brain, that's transmission. And here we can see that there are cranial nerves that are doing that. Nerves basically found in our head that take that information from the tongue and send it up through the brain. So notice that they carry information to the brainstem, the pons, and then the thalamus, which is the relay center for sensory information. And lastly, signals are relayed from the thalamus to, for example, the gustatory cortex. And that's where processing happens. We can say, this tastes sweet, this tastes very sweet, this tastes salty, so on and so forth. So we saw this figure before in chapter 28, but now we're going to make more sense of it. So as we know, different areas of our brain process different stimuli, resulting in our perception. Specifically, different areas of our ever so important cortex are involved in sensory processing. So we can see the gustatory cortex. cortex, for example, pictured here in pink, and we'll see the visual cortex, the auditory cortex, the olfactory cortex play a role in some of the other senses that we talked about. All right, so now that you have seen how taste is perceived, you can fill it out in your Chapter 29 worksheet on page 3. I recommend try and fill it out as you go, but also printing a second copy to rewrite this information once you've gone through the entire presentation and started to notice patterns. But it's good to take notes while you're doing it as well. So one more time, we'll go through taste. When we eat something sweet, like chocolate, for example, the sugar chemicals will bind to and stimulate specific taste buds, and that's reception. This will create voltage changes in the receptor cell, opening sodium ion channels, allowing sodium ions to flow into the cell. This changes the receptor potential, resulting in neurotransmitters being released to the sensory neuron. That's transduction. Those sensory nerves will transmit this signal all the way to the brain, specifically to, like, the gustatory cortex. That's transmission. The gustatory cortex will interpret these signals, process these signals, and then we perceive that we're eating something sweet, that we're eating chocolate, and that's kind of the processing step. Another thing about sensory perception we need to consider is how we perceive a variety of different stimuli. So we know that the rate of action potentials, how frequently they're fired, codes for the intensity of a stimulus, how sweet it is or is, how loud a sound is, how bright a light is. But how do we perceive a range of stimuli? By range, I mean how do we distinguish different flavors, sweet, salty, bitter, and beyond? How do we perceive different colors, different smells? The answer is we have different sensory receptors. So if we look at just taste, there are different taste receptor cells. So let's look at the taste bud that's pictured here. We can see that there are different receptor cells represented by different colors. So humans can distinguish at least 5 basic tastes, including sweet, salty, umami, bitter, and sour. And that's because we have different receptor cells that bind to different types of chemicals to create these different perceptual experiences. Umami, FYI, refers to savory flavors like broths, meats, things like that. And recently, lipid sensors have been identified on the tongue, which suggests that fat can be considered as the 6th taste, and so that'll be an interesting thing to learn more about as more research is done. So let's look at the taste bud again. So we have thousands, like around 10,000 of which found on our tongue. And if we just focus on 2, the one for sugar and the one for salt, we can see that there's a sugar receptor cell that responds to sugars and a salt receptor cell that responds to salt. Sugar and salt are 2 different chemicals, 2 different ligands, so they bind to 2 different receptors. Neuron transmission right now, sugar or salt, wild to think stimuli thing where it can diminish after constant exposure to it, kind of like with what we talked about. Therefore, Youll process stimuli when we humans that detect different types of receiving sensory stimuli. So there are 5 types of sensory receptors in humans that detect different types of sensory stimuli. Chemoreceptors detect chemicals, pain receptors detect painful signals, thermoreceptors detect temperature, mechanoreceptors detect changes in pressure that relate to things like touch or vibrations, and photoreceptors detect energy of light. So you've heard of at least some of these receptors before in class. For example, with chemoreceptors, we heard about them before in blood vessels that detect changes in pH. So hopefully you remember from Unit 2, we talked about how information from chemoreceptors in blood vessels is sent to our brain, and then the brain responds to this and sends signals out to our diaphragm to influence our ventilation rate. We talked about how stretch receptors in our stomach respond to the presence of food and initiate different types of hormonal responses. So stretch receptors are technically mechanoreceptors. Mechanoreceptors in our stomach stretch when food is present, causing them to send signals to the brain. The brain then sends signals back to the stomach, causing the stomach to release gastrin and acids to aid in digestion. We also talked about stretch receptors in our muscles, and we saw how these mechanoreceptors in the tendon of our knee influence our knee-jerk response. That said, we have heard about some of these, we can understand kind of what they do, but we're going to focus on taste, like we just did, hearing, vision, and smell. So we're really going to be looking at specific types of chemoreceptors, mechanoreceptors, and photoreceptors in this chapter, and how they result in our sensory perception. So the next sense we're going to look at is our sense of hearing. All right, so as we know, the ear deals with our sense of hearing, and it consists of a hearing organ. And so inside this organ, as we'll see, are mechanoreceptors that are capable of receiving sound waves. So before we get into the actual mechanoreceptors, we need to familiarize ourselves with the anatomy of the ear to see how sound waves travel from the environment into the inner ear. This should expand on what you looked at in lab. So the ear has three regions, the outer, middle, and inner ear. And the outer ear contains the pinna, which is the part we're most familiar with, like the part that has piercings. And it also contains the auditory canal. The outer ears function to essentially collect sound waves and channel them into the next part of the ear, the middle ear. In the middle ear, we see the eardrum. And the eardrum is a thin sheet of tissue that serves as a barrier between the outer and middle ear. So let's zoom in on the middle of the ear. When sound waves strike the eardrum vibrates, much like a drum getting struck by a drumstick. And it's these vibrations that are then passed into the inner ear. So the vibrations pass to three small bones and then to the inner ear. So we see the three small bones here, the hammer, anvil, and stirrup. You don't need to know them. Just know that there's a series of steps where the sound waves move from the outside of the ear to the inner ear. So our inner ear consists of fluid-filled chambers. One fluid-filled chamber is called the cochlea. The other is called the semicircular canals. So sound vibrations cause the fluid in the cochlea to move. So what we really want to look at when we're talking about sound perception is what's going on inside the cochlea. So let's zoom in on that. So the cochlea is the part of our inner ear that contains our actual hearing organ, which is called the organ of corti. And the cochlea looks a lot like the snail of a shell, and it contains the mechanoreceptors for transduction of mechanical sound waves into electrical signals in the form of receptor potentials and active potentials. So we're taking this mechanical energy, this sound wave, these sound waves really, and transforming that into electrical signals, which is like ions moving, right? So zoom in on the cochlea to see how reception and transduction actually take place. All right, so inside of the cochlea we find the organ of corti, which again is the actual hearing organ. But what we really need to look at is what it's composed of. It's composed of hair cells, which are the mechanoreceptors. receptors implicated in hearing. So in order to really understand how hearing works, we need to understand how hair cells work. We need to understand how sound travels from outside in the environment. Into our inner ear to affect the hair cells. So that's what we've been talking about now. But to actually understand reception and transduction, we need to look at the hair cells themselves. So pictured here is a hair cell. They're not actually hair, they're not related to hair. It's just that their structure has hair-like extensions. So the receptor cell, like we can see here, has hair-like extensions, and it's these extensions that respond to the vibrations caused by sound waves. So in our inner ear, in our cochlea, is some fluid, which we can see depicted in yellow here. When sound waves travel through our ear and reach the inner ear, they cause this fluid to move in a wave-like fashion. And this movement of fluid bends the hair cells in one direction or the other. When the hair cells bend, their plasma membrane stretches. So sound waves bend the hair cells in our ears, and that causes tension in the plasma membrane of the receptor cell, such that membrane permeability is altered, and we see potassium ions flow into the cell. So what happens is that mechanical energy that deals with movement or sound waves or the bending of these mechanoreceptors, is transduced into electrical energy. Mechanical energy is transduced into electrical energy in the form of a receptor potential. So we see cations move into the cell. This depolarizing signal results in the transduction step. So the depolarizing signal results in a release of neurotransmitters, which then influence the sensory neuron, which we see pictured here. The sensory neuron receives those neurotransmitters and fires action potentials, which then get carried to the brain. So now we have a better understanding of this mechanoreceptor and how it receives and transduces auditory stimuli. So this is a key part. Hearing, it occurs via mechanoreceptors called hair cells. So if we go back to what the inner ear looks like, we can see the organ of corti here, which we know is composed of hair cells and fluid. We're not going to get into all the details because this is a Bio 2 class, not a neuroscience class, so just whatever I'm telling you is what you're responsible for. So the hair cells in the organ of corti bend in response to vibrations and fluid movement. When they bend, signal transduction occurs because ions flow into the cell, and as a result, excitatory neurotransmitters are released. Sensory neurons respond to these excitatory neurotransmitters by firing action potentials that are transmitted to the brain. That's signal transmission. The auditory nerve refers to a bundle of sensory neurons that carry hearing information from the cochlea to the brain. And so we see the auditory nerve here, we can see it's a bundle of sensory neurons. So I want you to pause the lecture and watch the video on the next slide. It's in your PowerPoint to kind of visualize what we've been talking about. And then after doing that, see if you can fill out the 4 steps of signal transduction. So when we are filling out the steps that result in hearing, we need to start with the stimulus. We know that the stimuli are sound waves. We know that reception is done by mechanoreceptors, particularly hair cells, which bend in response to the vibrations. As vibrations are transferred through the ear, from the pinna to the eardrum to the ossicles to the fluid of the cochlea, they make the hair cells bend. When they bend, ion channels open, cations flow in, causing a change in receptor potential, like we've discussed. This depolarizing signal results in neurotransmitter release. The sensory neuron receives these neurotransmitters and fires an action potential. Many sensory neurons fire action potentials, and it's the collection of these sensory neurons that's called the auditory nerve. The axons of these sensory neurons conduct the action potential to the brain, and the information travels to the thalamus and then is relayed to the auditory cortex where it's processed so we can make sense of the sound and perceive it. So hopefully when you tried it out for yourself, it looked something like this, and hopefully you're noticing that it looks pretty similar to what we saw with taste. And so here we can see that auditory processing happens in the auditory cortex, which is a part of the temporal lobe. So you can pause the lecture now to try and fill out the worksheet if you'd like. Alright, so now that we're familiar with the steps of auditory perception, we're just going to add a couple extra things to what we know about our sense of hearing. So we saw that hair cells bend, to that is the release. Influences the baseline and C opposite direct. So in this way, how loud it is of the ear to conduct sounds. And this can be temporary, like when you have an ear infection, it can be permanent, pathway that can damage corti implants, which is nerve. Many living things evolve proteins that are capable of absorbing light, and we've seen these throughout the semester. So we've seen proteins called pigments that are able to absorb light. And these proteins are found within different structures in different organisms that sense or respond to light. For example, a euglena, which is a plant-like protist, they have eye spots that we see in red here that allow them to respond to light. Simple animals like flatworms can detect light in very basic ways using their eye spots, which are more advanced. And then with the evolution of more complex animals came the development of more complex ways of detecting light, such as eyes. And we read about this in lab and we saw it in the video. So like insects have eyes with many lenses, cephalopods, invertebrates like us have what's called a single lens eye. And that's what we're really going to be focusing on when we talk about vision, our eye, the human eye, and how we focus light energy on the photoreceptors and receive that information. So you should recall seeing this picture from lab, and we're going to quickly go over some important structures related to visual perception. So the very front of our eye is called the cornea, and it focuses light. It holds about 70% of the eye's focusing power. Inside of that, we have the iris, which is the colored part of our eye, blues, greens, hazels, browns, things like that. And the iris controls the size of the pupil, so it allows more or less light into the eye at a particular time. So the iris surrounds the pupil, which we can see here, and the pupil is the opening of the eye, opening in the center of the eye that lets light into the eye. So we need all these different things to be functioning in order for us to focus the light energy onto the retina. So after going through the pupil, light passes through a single lens, which we can see here in the light blue, hence our eye is called a single lens eye. The lens, which is that large disc behind the pupil, further focuses the light onto the retina. The retina is at the back of the eye, and we can kind of see it here in yellow. So this is where the photoreceptors are located, which we learned about in lab. So really, the first step of visual perception, reception, doesn't occur until the light reaches the retina. All the other eye structures are really just helping get light from our environment, focus onto the retina, focus onto the photoreceptors. And that's kind of similar to what we saw with the ear. All the other ear structures help to get sound wave vibrations to the organ of corti, to the hair cells. All right, so when the photoreceptors receive the light, they transduce light energy, light waves into electrical signals. And then sensory neurons send action potentials to the brain via the optic nerve. So the optic nerve is a bundle of sensory neurons that carry visual information from the eye, from the retina, to the brain. And hopefully you're now starting to notice a trend amongst our senses. We saw the auditory nerve, we saw cranial nerves carrying taste information. So these nerves carry these action potentials to our brain. So before we get into the 4 steps of visual perception, we're going to look at how light in the environment is focused onto the retina. And to do so, we need to look at the lens, which is something you got to hold and dissect in lab. So the lens is this flexible disc, and it is surrounded by muscles that help it move, that help change its shape in order to focus light. So we're not going to get into physics, we're going to keep it as simple as possible, just understanding that the lens needs to change its shape to focus on an object in our environment. So the lens is pictured here in light blue again, and it is connected to ligaments, which we see in blue and white stripes. And those ligaments are connected to muscles, which are in pink. So depending on if the muscles, the ciliary muscles, contract or relax, the lens is going to change shape. And in this way, we're able to focus on distant objects or nearby objects. So we'll look at what happens when we need to focus on something far away. So when the eye focuses on a distant object like a stop sign or a star, the ciliary muscles relax. So to imagine what it means when they relax, imagine that they like chill out, they stretch out, they kind of move away from each other. So the top and bottom ciliary muscles in pink move away from each other. They're kind of just like lounging back, if you will. So when they pull away from each other, that puts tensions on the ligaments, or pulls the ligaments, which causes the lens to stretch or flatten out. So when we try to see something far away, our lens flattens to focus the image on our retina, and that's what we see pictured here. When we need to focus on a nearby object, such as when we're reading words on a page, the ciliary muscles do the opposite thing. So the ciliary muscles contract, and what I mean by this is that they move closer to one another. When the 2 pink ciliary muscles that we see here move closer together, that puts slack in the ligaments. The ligaments become looser, they slacken, there's no tension on them. As a result, there's no tension on the lens kind of rounds, it becomes more round. So when we need to focus on something nearby, our lens rounds to focus the image on our retina. So once the light is focused on the retina, we can start to look at the 4 steps of perception. And so to understand reception, we need to look a little bit deeper into what are these photoreceptors. So there are 2 types of photoreceptors in our eyes. Rods and cones, that's what they're called. Rods let us see black, white, and gray. They let us see in low light conditions. They give us night vision. Cones let us see colors. They help us distinguish one color from another, and they play a role in our day vision. And so in this picture here, you can see how rods and cones are distributed in our retina. So this is our retina, kind of pictured as a circle, if you will, and the different rods and cones. And we can see that cones are centrally located, kind of blue, red, and green centrally located, while the rods are more so located in the periphery, and that's kind of the white dots that we see everywhere else. So interestingly, humans have more cones than rods because we are not nocturnal, we're diurnal, which means we're awake and most active during the day. So we've evolved more cones because they provide a survival advantage. They allow us to see in the day. An owl, or for example, which is nocturnal, or other nocturnal organisms, vertebrates, they're going to have few to no cones, but they'll have a lot of rods, so they have exceptional night vision. So rods and cones are photoreceptors, and that means that they're a specialized type of neuron that's capable of responding to light. And so here we can see the axon or synaptic terminals, and we can see the cell body. So they look like a neuron in that respect. However, they have a unique structure for their unique function. So rods and cones contain different pigment molecules that allow them to absorb different wavelengths of light, different types of light. So you can kind of see their structure here, and you can see where the pigments are located that respond to the light, and in response to that light, they send signals to the cell body, to the synaptic terminals, and that's kind of how reception and then transduction occur. So rods have a pigment called rhodopsin, and cones contain pigments called photopsins. There are 3 different cones, each with different types of pigments that absorb different wavelengths of light. So we say that there are red, green, and blue cones, like we see in this picture, and they respond to particular wavelengths of light. So let's say we're looking at an orange light. That's going to trigger a unique pattern of stimulation among these 3 cones. And this unique firing pattern results in our ability to perceive a wide range of colors. We don't just see red, green, and blue. We can see a wide variety of colors depending on the signaling pattern that results. Last thing we'll talk about with regards to these photoreceptors before we get into the 4 steps of perception is what's going on with color blindness. So color blindness results from a deficiency in one or more of your cones. So you might remember from lab or even bio 1 that red-green color blindness is an X-linked recessive trait, which means that people with this condition with red-green color blindness either have a defective red cone or the red cone is missing entirely. What does that mean? So the gene encoding the red cone, the gene encoding the pigment protein found in the red cone is found on the X chromosome, and the recessive allele is mutated such that the gene does not code for a functional pigment protein. As a result, their color perception is altered. And this condition is more common in men, biological men, because they only have 1 X chromosome. If they happen to receive an X chromosome with the recessive allele, they will experience color blindness. Biological females, on the other hand, have 2 X chromosomes, so they're only going to experience color blindness if they happen to receive the recessive allele from both mom and dad. So that's less likely to happen than if you just are relying on 1. So this happens more often in men. Alright, so now let's get into the 4 steps of perception. We have photoreceptor cells located in our retina, and we know that the retina is at the back of the eye. So pictured here is the retina made up of sensory neurons as well as photoreceptors. And in lab, you learned that our eye is wired backwards, meaning that light actually travels through various retinal cell layers before reaching the photoreceptors, because the photoreceptors are located at the back of the retina. So visual information, light waves, need to travel from the front of the eye all the way to the back of the eye to the photoreceptors. That's where the information is received, and then it's transduced in a way that it's moving towards the front of the eye. So that seems kind of strange, but that's how it works. Light travels to the back of the eye, that's where the pigments are, that's where it's received, and then transduction occurs and moves things kind of towards the front of the retina. Alright, so now that we have the photoreceptors down, let's go through these 4 steps of perception. When light comes into the eye, it focuses on the retina. The light interacts with the rods and cones, which are capable of receiving light energy due to the presence of pigment proteins. When rhodopsin and photopsins absorb light, they undergo a chemical change. They change chemically. That chemical change alters the permeability of the cell membrane, resulting in a receptor potential, a change in receptor potential. So a chemical change in these pigment proteins causes ions to flow. The change in the receptor potential results in the release of neurotransmitters from the axon terminals that are pointed out here with the black arrow. This should sound familiar because it's the same types of steps we've seen in taste and hearing. So we see a receptor potential change, we see neurotransmitters released from the axon terminals to affect sensory neurons. So in transmission, the sensory neurons respond to the neurotransmitters by firing off action potentials. Notice, though, that there are numerous synapses between the photoreceptor cells and the sensory neurons and among the sensory neurons themselves. So pictured here, we can see there's a lot of different synapses. One neuron can affect many different other neurons, or one photoreceptor cell can affect many sensory neurons. So these neurons in the retina are integrating information they're receiving from the rods and cones, and this integration that's happening just within the retina helps us with contrast and sharpening the images that we see. Once these signals get to the last neurons, we see that the axons get bundled together to form the optic nerve. So the optic nerve, which is basically a collection of axons, a collection of these sensory neuron axons, travels through the face and head to the brain. And specifically, the optic nerves carry signals to the thalamus and then the visual cortex. So in the processing step, the visual input from the eye is processed in the visual cortex and beyond, and we're able to make sense of the light stimuli in our environment. And as a result, we see things. So you can pause the lecture now to try and fill out your worksheet with regards to visual perception if you'd like. All right, so the last sense we're going to look at here is our sense of smell, and our sense of smell depends on chemoreceptors like we saw with taste. All right, so as a reminder, with taste, we saw a sweet chemical like glucose bind to its receptor, and the signal was transmitted via sensory neurons, and processed in the brain. And we saw that we have different taste receptors for different chemicals, such as sweet, salty, umami, bitter, and sour chemicals can be received by these different types of receptors. Your sense of smell also depends on chemoreceptors, which are receptors in your nasal cavity that respond to chemicals.
Okay, so I'm just gonna pause it right here. This particular part is actually the part that gets damaged when you have COVID and you lose your sense of smell, which just recently happened to me. Those neurons that are in your nasal cavity get damaged, and there's also some evidence that suggests through those axons are able to cross the blood barrier that protects your brain, and that's how you can, which I have, seemingly be able to encounter brain damage from COVID. So not just it's already brain damage when your sensory neurons are destroyed or damaged by COVID, and that's before it crosses into the brain. But you can have further damage if those viral particles actually get into your brain, which they can. So I've been recovering from COVID for, I'm starting to lose track now, I think 2 weeks. It sounds about right. Maybe I'm going on the 3rd week at this point. I don't know. But anyway, sorry, back to the lecture. I just wanted to point that out, that's what gets damaged and why people lose their sense of smell.
chemicals that smell, that are like odorants. So chemicals that smell we usually call odors, but what we're really talking about are chemicals in our environment that bind to receptors in our nose. So we cover smell last because it's slightly different than the other sensory pathways that we've looked at. So chemicals in the air that dissolve into the mucus that coats our nasal cavity are then going to be received by chemoreceptors. These chemoreceptors that are responsible for smell are called olfactory receptors. But the unique part here is that the olfactory receptors really are just sensory neurons. There's no distinction between receptor cell and sensory neuron. They're one and the same here, so you'll see reception, transduction, transmission are all done by the same cell. And that's different than what we saw before, where we saw a receptor cell that synapses onto a sensory neuron. So we're looking at kind of just one neuron here instead of two or more really. So if we look at the sensory neuron, we see that it has one end that extends into the mucous membrane of the nose and one end that extends into the brain. The mucous membrane of the nose is kind of in red here, and the brain is kind of in yellow here. So olfactory receptors are essentially the receptor proteins that receive the odorous chemicals, and these receptors are found on the dendrites of the sensory neuron. Specifically, they're found on the hair-like extensions, the cilia, of the sensory neuron that are extended.
Okay, so from this image right here on the left, you can actually see what I'm talking about. So you have the sensory receptors, the neurons, and see how they actually I don't know how to say. There's a canal. There's pathways from the nasal cavity into the brain. The reason that I point this out is because there's been a lot of in early COVID discussion of how it can possibly be crossing the blood barrier or the blood-brain barrier, which is that protective barrier. It's supposed to prevent viral activity and germs and that type of thing from getting in there and infecting your brain. But it has entry points from the spinal cord and the which connects to the medulla oblongata and up to the cerebellum. So this right here, you can see those pathways that connect. So those are the two entry points that COVID has to get into your brain.
Into the mucus. So people have about 450 different types of olfactory receptors that allow us to distinguish a variety of different tastes. For comparison, dogs have about 2 times as many olfactory receptors, so they can smell a lot more than we can. And when we're talking about an odorous chemical, one chemical can activate several different types of receptor, and it's that unique pattern of firing that results in our unique smell perception, something we've seen before. All right, so let's go through the 4 steps of olfactory perception. First is reception, in which odorous chemicals bind to olfactory receptors of the sensory neuron. They bind to these particular proteins. They bind to the sensory neuron. Once an odor molecule binds to a receptor, a depolarizing receptor potential occurs. Such that an action potential is fired. So here we're not seeing a neurotransmitter release, we're actually seeing an action potential fired. The action potential travels down the axon to the olfactory bulb of the brain, and synapses.
I'm actually going to take a photo of this so I can send it to my mom. Because I was trying to explain this to her the other day and she didn't understand what I was saying. I think it was my mom. Maybe it might have been somebody else.
With other neurons here. So the neurotransmitters released from the sensory neurons influence action potential firing rates in the olfactory bulb. So neurotransmitters released from the sensory neurons influence action potential firing rates in the olfactory bulb. And the olfactory bulb is just this region of the brain involved in smelling. All right, so from the olfactory bulb, signals are carried to the thalamus and the olfactory cortex, as well as other brain regions. The brain integrates these signals and makes sense of it so that we can perceive smells. The integration of information from hundreds of different receptors, plus the unique firing patterns, results in our ability to distinguish lots and lots of different smells. And it was recently estimated that we can detect around 1 trillion different smells. So even though we don't have that many receptors, it's this unique firing pattern that results in our ability to detect various different smells. One other thing to be aware of when we're talking about processing, yes, it's important to look at the auditory cortex, the visual cortex, the olfactory cortex, but it's also important to be aware that information also travels to places like the amygdala and the hippocampus so that we can associate smells, sights, sounds, and tastes with emotions and memories. So there's a lot of things happening in the brain at once to give us our perception, and we're just kind of scratching the surface. But hopefully you find it all very interesting. You can pause the lecture now to try and fill out the worksheet if you'd like. So with that, we have now covered how we taste, hear, see, and smell things. Make sure to fill out the worksheet so you understand the 4 steps of perception for each one of these senses, and you synthesize this information. Hopefully as you do that, you notice the patterns and see that.
Okay, so that's pretty much the end of that particular lecture. This is the last lecture, chapter 30. So, whoa, that's kind of off-centered, isn't it? Let me fix that. What the hell? Okay, so I know we have 13 minutes until the timer goes off, almost 14 minutes. I'm gonna go ahead and take a break since I'm going to the next one, and then the next one's only 30 minutes, and then I'm gonna start doing flashcards after that. I might stop the stream and restart it under a different title since I'll be doing flashcards at that point. But yeah, so I'm gonna take a break, and I'll be back. If you're studying with me, please go ahead, take a 13-minute break, then come back, or longer if you need to, whatever. But yeah, I'll be right back. Thank you, Pizza Man. Yeah, I need viewers. Actually, I don't mind. I don't study. Well, maybe you should. You should study something. It doesn't have to be school stuff. I'm still gonna have study with me videos after this because I'll be studying Japanese. I have found that doing this, I tried it out yesterday because I had not planned on doing like a video of me because I'm self-conscious. But I am, and the reason why is because since I know people are going to be coming in and out, like I have to be at my desk and I have to be studying, so it's holding me accountable right now to do the live stream this way. Yeah, thank you for the thumbs up. All right, so yeah, I'm going to take a break. I'll be back. Feel free to stay around. We'll be doing muscles next. Guys like muscles, right? That it might be helpful for you. Change the category. I have it listed as education, or at least it's supposed to be. That's how I filled it out. Change the title. What is the title? Oh my god, is it still snake.io? It's supposed to not be that. It's supposed to be study with me finals week. That's what I did. Did it not work? That's weird. Anyway, whoa, what's going on? There we go. Yeah, but so I'll be right back. Actually, what does the title say it is right now? I could stop the stream and then restart it when I come back if it's wrong. I'm checking. Let's see. Yep, it says snake.io. Damn it. Okay. Well, I had changed it on Restream. I don't know why it didn't correspond. It's probably correct on YouTube and not on Twitch, which sucks. So yeah, I'll go ahead and stop the stream, take a 10-minute break, and then I'll come back. And hopefully when I redo it, it'll fix it. But Thanks for telling me that. I appreciate it. All right.